Okay. Okay, very good afternoon to all the dignitaries of today's program. So, on the banner of CPGET Telangana, in association with the prestigious institutions in government degree colleges, that is Government Degree College for Women Jagityal, Government Degree College for Women Karimnagar, and one of the historical and prestigious institution in Karimnagar, SRR Government Arts and Science College, along with the another prestigious women's degree college in Twin Cities, that is RBVRR Women's College, Hyderabad, Autonomous. These institutions are going to start with one of the eminent and academic excellence faculty. We're going to start the CPGET-2023 PG entrance botany plant sciences online. So in this regard, we will going to start the inaugural program. So we wholeheartedly invite you, all the dignitaries, along with our today's patrons, Dr. Yamswani Satyanarayana Sir Garu, Principal, Government Degree College for Women Jagityal, my young and dynamic principal, and the SRR Government Arts Science Principal, Dr. Kalvakunta Ramakrishna Sir Garu, T. Sri Lakshmi Madam Garu, Government Degree PG College for Women Karimnagar, and Dr. J. Achita Devi Madam Garu, RBVRR Women's College, Autonomous Principal Madam Garu, and the today's program chief guest and hat-trick leader of our today's program, Dr.KADARU Surendereddy Garu, CPGET, and my godfathers are here. One is Professor T.Christopher Reuben sir, my professor, my godfather. The another one, the none of the other than Professor V.KRISHNAREDDY Garu, adjunct professor, Kakatiya University, Varangal, Department of Botany, and along with this eminent professor, retired and correspondent of RBVRR Women's College Autonomous, Professor Sudarshan Reddy Garu, and Head Department of Botany, RBVRR Women's College, Dr. Rajni Madam Garu, Kavita Madam Garu, and Head Department of Botany, SRR Government College, Dr. T. Shankar Sir Garu, and Head Department of Botany, Government Degree College for Women Jagityal, Sri G. Chandraya Sir Garu, and all the eminent botany professors and botany lecturers and resource persons, faculty, wholeheartedly welcoming you. At the same time, all the participants of today's program, wholeheartedly welcoming you. And Kavita Madam Garu will start the session and continue the program.
Okay, sir. Thank you very much, sir. Good afternoon to all the dignitaries. I, Dr. A. Kavita, Assistant Professor, Department of Botany, RBVRR Women's College. I welcome you all for this inaugural session of National Online Coaching in Plant Sciences, TSPSC 2023, organized by the 4 prestigious colleges of Telangana State, that is Government Degree College for Women Jagityal, Government Degree College for Women Karimnagar, RBVRR Women's College Autonomous Hyderabad, and SSR Government Degree College for Arts and Science Karimnagar. I'll start the program with a quote: "Successful and unsuccessful people do not vary greatly in their abilities. They vary in their desires to reach their potential," by John Maxwell. With this inspirational quote, I would like to welcome today's chief guest, Dr. K. Surendereddy Garu, General Secretary, TGCGTA, Telangana. I take the privilege to welcome the most distinguished and eminent personalities of plant science, Professor T.Christopher Reuben sir and Dr. V.KRISHNAREDDY sir, Professors, Kakatiya University, Varangal, Hyderabad. I also welcome all the principals of the 4 prestigious colleges and HODs of Department of Botany of all the 4 institutions. I welcome all for this inaugural session once again. Now I would like to call upon I request Dr. Surendereddy GARU, General Secretary TGC GTA Telangana, to speak few words on this inaugural session. Over to you, Surendereddy sir.
Madam, before message of brief message about the Surendereddy sir. Please, we will. Okay. So first of all, a brief CV I will going to present about this sir. I will present about the brief CV of our today's chief guest. So Dr.KADARU Surendereddy sir garu has previously worked as a teacher in Raikal Mandal, Jagatyal district, School Assistant Biological Science, Raikal Mandal, JDPHS Bhupatipur, and afterwards sir has worked as a junior college lecturer 14 years, and now sir has working in degree college as a assistant professor since 10 years, and his research experience is 8 years, and sir has published reputed journals, national 4 and international 4, and sir has attended seminar more than 20 national level and 20 international level. And sir has attended the refreshment course, 3 refreshment course in prestigious universities, and 1 orientation course, and 2 faculty induction programs and faculty development programs. And sir life philosophy is honesty, hard work, and humanitarian. And his success formula is: do not leave anything until it achieved. Do not leave anything until it achieved. Sir has his contribution to the department and also association. Sir now has recently has won in TGCGTA general elections. In 3rd time, hat-trickly, sir has won as a general secretary of Telangana Degree College Lecturers Association, just because of his commitment, dedication, determination, and that's why sir has enormously shown his capabilities regarding of today's scenario. Sir is only one and only the person who continuously his efforts to solve all the problems of Degree College Association. So really, we are wholeheartedly welcoming you, sir, and your services is always inspirable and appreciable. So thank you very much, sir. Please welcome and give a brief message regarding of today's program. Dr. Surendereddy sir garu. Okay, sir, please. Sir, your voice is not audible. Okay, sir. Sir, please unmute yourself.
Thank you. Thank you, Tirupati garu. Mute lo bettaram.
Sorry, sir.
Thank you.
Sir,
ఒక
welcome,
మంచి
sir.
థ్యాంక్ యూ మేడం, థ్యాంక్ యూ. ఒక మంచి కార్యక్రమాన్ని నిర్వహిస్తున్నటువంటి కన్వీనర్ తిరుపతి గారు, అలాగే జగిత్యాల ఉమెన్స్ కాలేజ్ ప్రిన్సిపల్ వై సత్యనారాయణ గారు, ఎస్ఆర్ఆర్ కళాశాల ప్రిన్సిపల్ రామకృష్ణ గారు, కరీంనగర్ ఉమెన్స్ డిగ్రీ కాలేజ్ ప్రిన్సిపల్ శ్రీలక్ష్మి గారు, అలాగే ఆర్బివిఆర్ఆర్ ప్రిన్సిపల్ అక్షితాదేవి గారు, ఈ కళాశాలకు చెందినటువంటి హెడ్స్ ఆఫ్ ది డిపార్ట్మెంట్స్ అండ్ ప్రొఫెసర్ కృష్ణారెడ్డి గారు, క్రిస్టఫర్ గారు, కరెస్పాండెంట్ ఆర్బివిఆర్ఆర్ కాలేజ్, అందరికీ నా యొక్క నమస్కారాలు. చాలా సంతోషంగా ఉంది. మన ఫ్యాకల్టీ మెంబర్స్ అంతా కూడా కేవలం మన కళాశాలలో చదువుతున్నటువంటి విద్యార్థులకి మన సిలబస్ కంప్లీట్ చేయడమే ఒక టార్గెట్గా, మన డ్యూటీగానే కాకుండా మన కాలేజీ నుంచి బయటకు వెళ్తున్నటువంటి విద్యార్థుల యొక్క భవిష్యత్తు కోసం, వాళ్ళ భవిష్యత్తు బాగుండాలని, వాళ్ళు వివిధ స్థాయిలలో నిలదొక్కుకోవడానికి వాళ్ళకి కావాల్సినటువంటి గైడెన్స్ ఇవ్వాలని చెప్పేసి ఈ విధంగా మనం కోచింగ్ అనేది, ఆన్లైన్ కోచింగ్ అనేది ఏర్పాటు చేస్తూ మళ్ళీ పిల్లలందరికీ కూడా అందుబాటులో ఉండే విధంగా దీన్ని గత 2-3 సంవత్సరాల నుంచి నిర్వహిస్తూ రావడం అనేది చాలా సంతోషకరంగా ఉంది. లాస్ట్ బిఫోర్ ఇయర్ దాదాపుగా ఒక 40 మెంబర్స్ మన విద్యార్థులంతా కూడా వివిధ యూనివర్సిటీలలో సీట్లు సాధించడం, లాస్ట్ ఇయర్ మోర్ దెన్ 30 స్టూడెంట్స్ సీట్లు సాధించడం అనేది నిజంగా మన ఫ్యాకల్టీ మెంబర్స్ అందరికీ కూడా అభినందనీయం. చాలా మంది, దాదాపుగా 25-30 ఫ్యాకల్టీ మెంబర్స్ వీళ్ళందరినీ కోఆర్డినేట్ చేస్తూ, ఇన్ని కళాశాలకు చెందినటువంటి రాష్ట్ర వ్యాప్తంగా ఉన్నటువంటి విద్యార్థులందరినీ కూడా కోఆర్డినేట్ చేస్తూ ఎప్పటికప్పుడు వాళ్ళకి కావాల్సినటువంటి సలహాలు, సూచనలు ఇస్తూ మరి ఈ కార్యక్రమాన్ని ఇంత విజయవంతంగా, ఇది బహుశా 3వ సంవత్సరం 4వ సంవత్సరం అనుకుంటాను, 4 సంవత్సరాలుగా ఈ కార్యక్రమాన్ని విజయవంతంగా నిర్వహిస్తున్నటువంటి నిర్వాహకులందరికీ కూడా నా తరఫున కృతజ్ఞతలు, అభినందనలు. అలాగే మన గవర్నమెంట్ డిగ్రీ కాలేజీలో పనిచేస్తున్నటువంటి అధ్యాపక మిత్రులందరికీ కూడా మరొకసారి మనస్ఫూర్తిగా అభినందనలు తెలియజేస్తా ఉన్నాను. మనందరం కూడా డిగ్రీ కాలేజీలలో పనిచేస్తూ మన వ్యవస్థ బాగు కోసం మనం చేస్తున్నటువంటి కృషి చాలా అభినందనీయం. ఎందుకంటే ఒకప్పుడు 29-30 వేలు ఉన్నటువంటి అడ్మిషన్స్ మన రాష్ట్ర వ్యాప్తంగా ఈరోజు 52,000, 53,000కి రీచ్ అయ్యామనంటే ప్రతి అధ్యాపకుని యొక్క పాత్ర ఉంది. మన గవర్నమెంట్ డిగ్రీ కాలేజ్ కూడా సొసైటీలో చాలా మంచి పేరు తెచ్చుకొని మంచి పురోభివృద్ధిలో కొనసాగడానికి ప్రతి అధ్యాపకుడి యొక్క పాత్ర కూడా ఉంది. ఆ రకంగా ఈ అధ్యాపకులందరికీ కూడా నేతృత్వం వహిస్తున్నటువంటి నాకు కూడా చాలా సంతోషంగా ఉంది. రాబోయే రోజులలో మరి గవర్నమెంట్ డిగ్రీ కాలేజెస్ కూడా మరింత ముందడుగు వేసే దిశగా మన ప్రయత్నాలన్నిటి సాగుతూ ఉన్నాయి. మనందరికీ తెలుసు, గడిచిన 2-3 సంవత్సరాలుగా యూనివర్సిటీలలోనే కాకుండా మన గవర్నమెంట్ డిగ్రీ కాలేజీలలో కూడా రీసెర్చ్ సెంటర్స్ ని మనం ఎస్టాబ్లిష్ చేసుకోవడం, తర్వాత చాలా మందికి మనలో ఫ్యాకల్టీ మెంబర్స్ కి గైడ్స్ కూడా రావడం జరిగింది. మరి ఆ రకంగా అన్ని కాలేజీలలో అన్ని సబ్జెక్టులకు సంబంధించి కూడా రీసెర్చ్ సెంటర్లు రాబోయే రోజులలో వచ్చే అవకాశం ఉంది. అలాగే ఆల్ ది ఎలిజిబుల్ ఫ్యాకల్టీ మెంబర్స్ అందరూ కూడా గైడ్షిప్ పొందేటువంటి అవకాశం కూడా ఉంది. దానివల్ల రీసెర్చ్ విషయంలో కూడా మన కాలేజ్ చాలా ముందుకెళ్ళేటువంటి దిశగా మనందరి ప్రయత్నాలు సఫలీకృతం కావాలని చెప్పేసి నేను కోరుకుంటా ఉన్నాను. అలాగే మరి ఈ సంవత్సరం కూడా మన కోచింగ్ కార్యక్రమం అంతా కూడా విజయవంతమే. చాలా మంది విద్యార్థులకి మన స్టేట్ యూనివర్సిటీలలోనే కాకుండా మరి సెంట్రల్ యూనివర్సిటీలలో కూడా మంచి ర్యాంకులు రావాలని, వాళ్ళు భవిష్యత్తు బాగుండాలని చెప్పేసి కోరుకుంటూ మరి దీంట్లో పార్టిసిపేట్ అవుతున్నటువంటి ప్రతి అధ్యాపక మిత్రునికి, దీన్ని నిర్వాహకులకు అందరికీ కూడా మరొకసారి కృతజ్ఞతలు తెలియజేస్తూ నాకు ఈ అవకాశం ఇచ్చినందుకు మీ అందరికీ కూడా అభినందనలు తెలియజేస్తూ ముగిస్తాం.
Sir, thank you very much, sir, for your inspirational speech on this occasion. Really, your insights and inputs can be added to our coaching, sir. Thank you very much.
Thank you.
Now, I would like to invite Satnarayan sir, Principal, Government Degree College for Women, Jagitial, to speak few words on this occasion and start the program. Over to you, sir.
Thank you, madam. Very good afternoon to all. Honorable visionaries and dignitaries on the screen and off the screen, and all the endeavors aspirants, a very good afternoon. I am Dr. V. Sachinarayana, Principal and Chairman of this CPGT, GDC Women's Jagitial, is cordially welcoming everyone to this prestige knowledgeable session on behalf of GDC Women Jagityal, and I welcome to all the participants and all the dignitaries in the meeting. Dr. Ramakrishna Garu, Principal, SRR Gas Autonomous Karimnagar, and Principal Madam T. Sri Lakshmi Madam Garu, Government Degree College Women Karimnagar, and Dr. A. Achyuta Devi, RBVRR, Women Hyderabad, and our chief guest, Dr. K. Surendreddy Garu, PGCPSC Garu, and Professor V.KRISHNAREDDY, Adjunct Professor KU, Department of Government, and Professor Christopher Garu, Dean R&D Department, Biotechnology, Kakti University, and Director T-Hub also, and Professor Sudarshan Reddy Garu, RBVRR, and our college Vice Principal, Dr. Masroor Madam, and our academic coordinator, Dr. A. Jyoti Lakshmi, and IKC coordinator, Dr. Harjot Kaur Garu, and HOD, Botany Department, G. Chandre Garu, and Staff Club Secretary, Dr. M. Satyaprakash Garu, and all my staff members. Let us begin the session with the brief introduction of the glorious program. Actually, this CPGET online coaching program has taken initiation in the year 2020 by our former principal, Sri M. Srinivas Reddy Garu, in the hottest pandemic period, with the collaboration of RBVRR, Reuben College, Hyderabad. Then 200 participants registered on that time, and 40 students get PG admissions in prestigious institutions, and 18 members secured below 100 rank, and it's been glittering the limestone in the crown of our GDC Women Jagityal, and it's been with the tremendous success since 2 years, and this year we are joining hands with the excellence to other institutions like SRR Government Arts and Science College Autonomous and Government Degree College Women Karimnagar to make it more success for the future endeavors. Around 340 members were registered for this academic year from the various states in our country like Telangana, Andhra Pradesh, Maharashtra, and Kerala. We hope surely this program will ensure all the future endeavors for all the aspirants with all success. Thank you for giving this opportunity, sir. Organizers. Thank you, sir.
Thank you very much, sir. Hopefully, we'll continue this for the next academic years also.
Yes, yes. Okay,
and we are wishing for the good luck for this year also, sir. Thank you very much.
Okay.
Now I would like to welcome Dr. K. Ramakrishna Garu, Principal, SRR Government Degree College, Arts and Science, Karimnagar. Sir, over to you, sir, to speak few words on this occasion.
Good afternoon. I am audible.
Yes, sir. Good afternoon, sir.
A memorable and happy moment to all the government college lecturers and TGC GTA association friends. Just now spoke my beloved friend, my classmate also, Dr. Surendra Reddy, and young dynamic coordinator, Mr. Padala Tirupati. He is the center of attraction. He is the coordinator. He is the vibrant person in plant science and biotechnology and botany. Our beloved friend Y. Satyanarayana Garu, Principal, GDC Women Jagityal, and GDC Women Karimnagar, Principal Sri Lakshmi Garu, and Chandraya Garu, Achyuta Devi Garu, Professor Christopher Garu, Professor Krishnareddy Garu, and all the other dignitaries, Rajani Garu, Shankar Garu, Parvati Garu, Padmaja Garu, and who are participating in the this inaugural session, National Online Coaching for CPGET-2023, Plant Science and Plant Biotechnology. It's a wonderful situation. wonderful platform to share the this platform. I hope Tirupati and his team very good coordinating and working. Just now you heard number of achievers in through this online coaching. 40-50 members are selected in various central universities and state universities through online coaching. I hope this type of coaching is very attractive and very informative and useful and very inspiration to all government degree colleges in the state. Not only limited to the these 3-4 degree colleges. It is already spread to all over Telangana. Tirupati is the center of attraction. Just now I said, and his team, Satyanarayana Garu and Masroor Garu and Harjot Kaur Garu, Women's Degree College, Jagityal team and all other plant science professors and lecturers are very well coordinating, very friendly. They are thinking about the future generations. Quality is not a destination, it is a continuous journey, saying in English. Like that, continuity is the most important. When Tirupati started this coaching program first time, never anyone expected to continue the same. It is not a simple issue. It's a manpower and networking and a subject division and continuity is the main important. These type of coaching platforms are very informative and useful for the young students, those who are aspirants of PG in various universities, central and state universities. I congratulate once again coordinator and all the dignitaries, professors and mentors of this program. I hope this program will be very inspiring and useful for the young scholars of the life sciences. I am very happy because our college is also part of this program. Continuously, I think this is the third time we are getting together, going for forward for the benefit of the future generations and students. Congratulations once again. Wish you all the success, all the best. Thank you, one and all. Thank you. Namaste.
Thank you, sir. Thank you very much for your valuable words. And I hope all these 4 institutions will go as far for getting a successful results for the students. Thank you very much, sir. Now I would like to welcome our Vice Principal, Dr. C. Kavita of RBVRR Women's College, to deliver a few words on this occasion. Over to you, ma'am. Ma'am, Kavita, ma'am. Hello. Sir, am I audible?
Yes, you are audible, madam.
Yeah. Kavita, ma'am. As our principal, Achita Devi ma'am is a little bit busy in her schedule. I invite Kavita ma'am. So there is something glitch in the internet, I believe, sir.
Sudarshan, sir.
Yeah. I invite our secretary cum correspondent G. Sudarshan Reddy sir of RBVRR Women's College to share few words, sir.
Yeah, good afternoon. Good afternoon.
Good afternoon, sir.
Thank you, Kavita.
Yes, sir.
This is a very happy occasion to be part of this program. Firstly, I welcome Dr. Surendereddy Garu and guest speakers, Prof.T.Christopher and Prof.T.Christopher Reuben, and the principals of the host colleges. 3 are located in Karimnagar district, 1 is in Hyderabad. And the coordinator of this program, Dr. Padala Tirupati, and my own colleagues in RBVR Women's College, Dr. Reddy, Dr. Kavita, and Vice Principal Kavita. Indeed, this is a well thought out program. This was mooted about 3 years back. I think this is 3rd year. This was mooted by, I think, Dr. Tirupati and Dr. Rajani to start during pandemic situation, I think in 2021, to start this online coaching program for students of plant sciences by coming together. Then slowly 2 other institutions have joined. And today I think this program is touching every part of Telangana state, and large number of candidates are making use of this coaching program. This is being offered on a very nominal fee and a very quality coaching, because as I have seen last year, in the first year, then last year, the very good and very serious faculty is being drawn, and a number of topics related to the entrance programs of various central universities and also CPGET of Usmania University. So then I think this program must go on, and this collaborative effort proved to be successful, and all the faculty members of the plant sciences from across Telangana universities and colleges are, I think, part of this program, offering very high quality coaching to the students located in different parts of Telangana state. And this is largely successful, I personally feel, because of the grit and determination of one person who is convener of this program, Dr. Padala Tirupati, supported by the faculties from across the universities and colleges from plant sciences department. So hence, this is in fact this online coaching, in fact, we all know is the result of pandemic situation, and this proved to be a successful. We are trying to make use of the technology successfully to reach out to the students of plant sciences located across Telangana state. And this is wonderful program. This program must go on, come what may. We are with Dr. Tirupati, and this program must go on. This program must reach the candidates in the entire Telangana state, and more, I am told good number of candidates are being selected last year and before last year. This year also, I hope large number of students make use of this coaching program and come out successfully. And I thank the principals of 4 institutions and the faculties and the faculty of various other departments and also universities. On behalf of RBVR Women's College, I thank one and all and for inviting me to be part of this program. Thank you very much. Thank you one and all.
Thank you very much, sir. This was a very good encouragement for us to go in our journey of this online coaching for this academic year, sir. So there was almost by till date more than 300 participants have registered for this program, sir. So that could be a very good number, and we
Yes,
are
of course.
Yes, sir. That is a very good number, we believe. So we could see this number in success rate will be the same.
Yeah,
We
good.
hopefully with your encouragement and support, we can achieve. We have achieved that in the past 2 years, and now seeing for the best for the next year, sir. Thank you very much, sir.
Thank you. Yeah.
Right. Now I would like to welcome Sri Lakshmi GARU, Principal, Government Degree College for Women, Karimnagar, to share few words on this occasion. Ma'am, are you available? Sir, ma'am is available.
Sorry, madam. Madam is not joined because of.
Yeah. Sir, you're on mute. Yeah. Okay, sir. Yeah. Now I would like, it's my privilege now to introduce today's guest speaker, Professor Krishnareddy, sir. Krishnareddy, sir, is a retired professor of Department of Botany, Kati University, Warangal. Sir has an educational qualification from Kati University, Warangal, right from 81 to 89. Sir completed his PhD with entitled Studies on Mycotoxin Producing Fungi Associated with Maize. Sir has published 53 research papers, both internationally and 36 nationally in reputed journals. Sir has guided 7 PhD students and currently guiding 4 students. Sir has delivered 42 invited lectures and presented seminars and papers. Sir has published 10 books. Sir is also a resource person for Telugu Academy, B.R. Ambedkar Open University, and Telangana State Council for Higher Education. Sir was held at various positions like Program Officer, Additional Co-Examinations, Additional Controller of Examinations, University Nominee on Governing Bodies, Head Department of Botany, and Chairman BOS in Department of Botany, Kati University. Sir is also a member of scientific societies like Indian Botanical Society, Indian Phytopathological Society, Society of Mycology and Plant Pathology, and Society of Indian Poultry Science Association. Sir is member of various academic bodies in holding BOS meetings and syllabus framing committee of well-prestigious institutions overall Telangana state. Sir, with this brief introduction, I would like to welcome you and deliver your valuable suggestions and talk on this session. I welcome you once again, sir.
Good afternoon to all of you. Madam, is it audible?
Yes, sir. Yes, sir. You are audible, sir. Please.
Okay. Good afternoon to all of you. So, Dr. Surendra GARU, General Secretary, TGC-GTA, who is today's chief guest of this program, and the respected principals of Government Degree College, Jagitya, Government Women's Degree College, Jagitya, Government Women's Degree College, Karimnagar, SRR College, Karimnagar, and RBVR Degree and PG College, Hyderabad, and my friend and my colleague, Dr.T.Christopher, who is the keynote speaker of today's inaugural session, and Dr. Padala Tirupati, convener of this program, and other organizers and subject experts from different parts of the Telangana. and participant students. At the outset, I am highly thankful to Dr. Tirupati, who was our student, for inviting me and making me a part of this very useful and important program. I congratulate Tirupati and also other organizers, his team, for making efforts to success this PG set entrance coaching program. I am also from this platform, I am congratulating Dr. Surendereddy GARU for becoming General Secretary successfully for the third time in the prestigious association, that is a TGC GTA. I am also congratulating all the principals of 4 colleges for their initiation to organize such type of program, because the colleges are located in different parts, and different parts, that is Jagityal, Karimnagar, Hyderabad. Students are participating from different parts, and the organizers are from Jagityal, and bringing all these different persons to a common platform is not so easy. They have to carry out a Herculean task to make this program success. And I also feel that the coaching program will also be success in coming days. Now, it is a very good program, and I appreciate all the organizers. They deserve the appreciation. The students are lucky because now this is a mainly free coaching with a nominal fee for the PG Common Entrance Test at the state and national level for state universities and central university. I feel that this is not only entrance test for the common entrance test for the botany, and this the coaching given here will be useful for various other competitive exams like Group 1, Group 2 exams, or IFS, or even for public service commissions also, Union Public Service Commission, because subject is same. Everywhere, wherever you teach, the subject is same. For example, you take the Krebs cycle in respiration. So Krebs cycle is taught in the intermediate, it is taught in the degree, it is taught in the PG. But the level of teaching and level of understanding is different at different levels. But basic thing, the substances as well as the enzymes involved are the same in the entire Krebs cycle. So the student got an authority on the Krebs cycle. He can answer the question in any form. He can write an essay, or he can write a short note, or he can answer in a form of the short bit or multiple choice questions. So like that, for example, take the glycolysis, C3 cycle, C4 cycle, biosynthesis of proteins. So for all these things, the basic things, the basic principles are same, the basic subject is same. So if the resource persons make the students understand the theme of these things, it will be very useful for them, not only during this time, even after getting seat, even after completing their MSc also, they can go for various competitive exams starting from JLs to UPPSC or APPSC, TSPSC exams, so that these subjects are always useful for them. Now, I advise all the resource persons to stress or to give more concentration on the subjects like genetics, cell biology, molecular biology, environmental science, plant physiology, biodiversity, rDNA technology, and molecular biology, like that, to give more stress on the subjects, from which you will get questions in all the exams, not only in the PG set, even other exams, other competitive exams also, the questions will be mainly from these subjects. So this, if you concentrate on these subjects, it will be more useful to the students. As you know, most of you know, there are nearly 45 to 50 colleges offering MSc Botany in our state. And the highest number of colleges and competitive prediction, there are 21 colleges. And I think Shatavan University 8 or 9, in Usmania a dozen colleges. And altogether there are 45 to 50 colleges that are offering this MSc Botany course. And of course, number of seats are there, but competition is also there. There are many students competing for these admissions. Now, the getting seat is not an easy task, and there is a heavy competition. This is the era of competition. So the student should aim not only to get seat in the PG, but also to get the knowledge in the subject, so that it will be more useful for him. Now, I hope the students will get a lot of information from the resource persons, because most of the resource persons are degree college lecturers. And Tirupati, what I advise Tirupati, you can also take the help from the university teachers in some of the topics. For example, Professor Reddy sir is there, Professor Sadanandam is there, Professor Mustafa is there, and there are some other, Dr. Sureka is there, Professor Sureka is there. They can also deliver some important lectures for your students. So with like this, during this competitive era, the students should get use of these experts. I also advise all the students to learn the subject in depth, because so once they get command on the papers or subject topics, it will be useful for them during their entire life if they settle in the teaching field. Now there are, for example, I want to recollect one thing. Dr. Chandrasekhar, one of our colleagues in the university, he is also giving free coaching to the MSc entrance examination for the last 3 years. During the COVID period also he has given, and many of the students were admitted. So like that, if I recollect back, in Warangal, the government Pingle College and also Kaktiya Degree College, they offered free coaching for chemistry entrance examination for several years. So now it has discontinued, because I think most of the senior teachers were retired from their services. But now what I observe is most of the resource persons are young teachers, they have a lot of service. And the coordinator like Tirupati is also young and has a lot of service. So they will, I hope they will continue this program for a long period, for several years coming, to benefit the students of botany to settle in the life. And also there are several books available in the Telugu Academy. So I advise all the resource persons to verify the books of Telugu Academy, because most of the books were written by our department faculty. That's myself, Dr. Christopher, Dr. Mustafa, Professor Sureka, and Dr. Reddy from KDC. And editors were Professor Reddy sir and Professor Reddy sir editor. And almost all the books are available in different branches of the botany. So those books were written with the latest information available also. So I request all of you to refer those books during this coaching period. So with these few words, I once again thank the Tirupati especially and all the organizers for giving me this opportunity. And I wish all the success for the teachers as well as to the students to get more seats. And moreover, I want to stress one more thing. Nowadays, 5 years back, situation was different. Now there are many residential colleges run by tribal welfare, social welfare, RC welfare, like that, and the cream students are coming. I was surprised to see in our MSc final year students, when we conduct seminars for MSc final year students, some of the students, they are teaching better than teachers. They are like teachers. They are preparing PPT well, and they are teaching like a perfect teacher. They are very cream students. So the students are, when the students are cream, when the students are meritorious, the teachers has to work more. So here I want to stress all the teachers who are involved in this teaching program, be cautious of the students, because in all the residential colleges, they are cream students, and in our department, Bharati Kaktiya University, we are getting very good students from the residential junior colleges. So with these few words, I wish all the best for the successful completion of this program. Dr. Tirupati, Madam, Rajini Madam, and other all madams for the, and also I wish all the best to all the guide teachers or for resource persons. So thank you very much for involving in this program. Thank you.
Thank you very
Thank you,
much, sir,
sir.
for
Thank
your
you, sir.
valuable
Thank you very much, sir.
suggestions. Hopefully we follow your suggestions, sir, of referring the books and guiding our students in the right pathway to achieve the goals and the results. Thank you very much, sir.
Okay, madam. Thank you. Thank you very much.
Now I would like to invite Dr. P. Manjula, Assistant Professor, Department of Botany, RVR Women's College, to introduce Christopher, sir. Over to you, Manjula.
Thank you, madam. Good afternoon, one and all. It gives me an immense pleasure to introduce today's guest speaker, Professor Christopher, Reuben's Department of Biotechnology, Chaitanya Deematuri University, Warangal. Sir has 15 years of teaching experience in UG and PG biotechnology, and Sir has 20 years of research experience in broad areas of plant tissue culture, genetic transformation, molecular markers, and Sir has guided EIT PhD students, and Sir has to his credit of publishing 48 research papers in international and national journals, and also authored 8 books. SAR has successfully completed 3 research projects, and SAR has given 25 invited lectures and has been a resource person in various conferences and seminars, and a member of various academic bodies. Sir received University Research Fellowship and Senior Research Fellowship from Kakatiya University, Warangal, and Research Associateship at University of Delhi South Campus, New Delhi. And Sir has held various positions like Head, Course Coordinator, Additional Controller, and Director, Foreign Relations Officers, and currently Dean Research Development, and newly appointed as the first Director of T-Hub, Kakatiya University, Warangal. So through this brief introduction, now I hand over the session to Sir. Thank you, sir.
Over to you, sir.
Yeah, thank you, madam. I want to use, you can make me co-host?
Tirupati, sir.
Yes, sir. Yes, sir.
Make
Please
the-
you share. Please share your screen also, sir. Please welcome, sir.
Yeah. Host disabled participant screen sharing.
One second. All right, sir. Now you are share your screen, sir.
No? Okay. Madam, can you see it now? Tirupati, కనబడుతుందా?
Yes, sir. Yes, sir. Screen is visible, sir.
Madam? Rajini, madam?
Yes, sir. Screen is visible, sir. Please you proceed.
Okay. First of all, I thank the principals of Government Degree College for Women Jagitial, Government Degree College for Women Karimnagar, RBVRR Women's College Hyderabad, SRR College Karimnagar, for arranging this particular national online coaching program for the benefit of students who wish to appear for PG-CET examination test that is going to be ensured in the coming days to come. First of all, actually, I want to thank first of all, I like to thank Surendra, sir. Unnara, sir? Hello?
Yes, sir. Yes, sir. Surendra sir is there, sir.
Sir, ఆయనకు దండం పెట్టుకోవాలి. I
Sir,
salute.
Surendra sir.
Hello.
Sir, namaste.
Sir, namaste, sir.
Sir, thank you so much. You have helped a lot of research, lot of junior college lecturers and degree college lecturers. జూనియర్ కాలేజీలో నా స్టూడెంట్ కూడా ఒకడు ఉన్నాడు సార్. మీకు సల్యూట్ సార్. ఎందుకంటే ఈ పరిస్థితిలో
పెద్దవాళ్ళు మీ ఆశీర్వాదం ఉండాలి సార్.
ఇంత వర్క్ చేసి వాళ్ళకి మీరు ఉద్యోగాలు ఇప్పించిందనంటే I salute, and you have given them life. So from my behalf, from my family, from my, Tirupati is my student. Krishnareddy మేము అందరం ఒక దగ్గర పనిచేస్తాము. మా botany department తరఫున మేము we are really grateful to you for your efforts that you have given life to so many people. ఇదే చెప్దాం అని అనుకున్నాను, అందుకోసమే మిమ్మల్ని ఉన్నారా అని అడిగాను సార్. సార్ థ్యాంక్ యూ సో మచ్ సార్, థ్యాంక్ యూ సో మచ్.
Thank you and welcome, sir. Thank you for your blessings.
Today I'm going to discuss on one important topic, that is in cell biology, eukaryotic chromosomes and also chromatin material. That will be the topic for the keynote address today: eukaryotic chromosomes and chromatin material. This is the slide that will give you the information about chief guests, co-patron and conveners. I also congratulate all the 12 subject experts who will be actually teaching, coaching students to appear for this particular entrance test. My special thanks to Dr.PADALA Tirupati, who has been instrumental in catching both of us, me and Krishnareddy, to participate or to be in this particular program. I thank you, everybody. So my first slide regarding eukaryotic chromosome it should be visible. It is derived from the word chroma, means colored, zoma means body, so it's a colored body, and chromosomes are localized in the nucleus, both in plant and animal cell. And if you want to visualize, if you want to see chromosomes under a microscope, they have to be stained. Staining is a must. On your left side, we have mitotic chromosomes being stained, and then you can see the chromosome stain. On the right side, you have a meiotic cell where the chromosomes are stained. Staining has to be done for visualizing chromosomes. Otherwise, chromosomes are not visible. We cannot see them. So you see a beautiful picture of mitotic chromosomes. This is stained by acetoarsine, while meiotic chromosomes are stained by acetocarmine. You can also call it as carmine. So in the exam, they might ask you, in the entrance test, they might ask you what are the stains commonly used to stain mitotic chromosomes. You can give the answer as orsine, and then for meiotic chromosome, it is carmine. Okay, right. So we'll first look into the history of the chromosome, how chromosomes were actually first discovered, observed. Okay, so in 1842, it is Carl Nageli. He observed thread-like bodies in plant cells. Then 1848, it is Hofmeister, who found in pollen mother cells some stained bodies of Tradescantia. He saw some stained bodies in that. And then in Strasburger in 1875, reported the presence of rod-shaped structures. 1882, Walter Flaming called it as chromatin material, and also he was responsible for identifying the phases of mitosis, that is your prophase, metaphase, anaphase, and telophase. Then in 1888, Waldier coined the term chromosomes to these rod-like structures because they had affinity to basic dyes. And then 1900, Sutton and Boveri proposed chromosomal theory of inheritance, which means to say that chromosomes are responsible to carry genes from one generation to the other generation. And in 1910, Thomas Sutton proved that the chromosomes carry traits. Traits are the characters that will be transmitted from one generation to another generation. So this is a brief history. So in the exam, they might ask you who coined the term chromosome. Then you should be able to tell them Waldier. Who proposed the chromosomal theory of inheritance? Then it should be Sutton and Boveri. So these questions could come in your entrance test. So please make a note of all this information that I wanted to present. Okay. So this is the drawing of Walter Fleming. He had this hand drawn pictures that are seen here. And this is Walter Sutton and Theodore Boveri. They are responsible for chromosome theory of inheritance. This is Thomas Hunt Morgan, who actually showed that characters are linked to chromosomes, or you call them as traits. Characters or traits are linked to the chromosome. In this picture, you see a bunch of banana are hanging here in his lab. This is his lab. This bunch of banana that we see here tells us that he was working with Drosophila. So you can see a very crude kind of microscope and small bottles here. And he used to grow Drosophila in these bottles using this banana. Even students also can grow Drosophila on banana even in your lab experiments also. So he has contributed a lot for the traits. This is the structure of a chromosome. You have a linear body-like structure with a centromere in the center and DNA running inside. And DNA, at the end of this DNA, you have telomeres. These ends are telomeres. And then you have a secondary constriction called satellite or trabant. And then DNA, only one piece of DNA runs through this. Okay, so this is a typical structure of a chromosome. And these are called chromatids. This is one chromatid, this is another chromatid. This is one chromatid, this is another chromatid. This is called NOR region, nucleolus organizing region, NOR region, where nucleolus is attached to this chromosome. And then this is the site where ribosomal RNA are also synthesized. So this is eukaryotic, typical eukaryotic chromosome, right? So this is what we have seen. And then based upon their position of centromere, in exam definitely they will ask you this question. Based upon the position of the centromere, chromosomes have been classified into 4 groups. First is called metacentric, wherein the chromosome is in the center of the chromosome, dividing it in 2 halves. Okay, and at the time of anaphase or metaphase and anaphase, the chromosome will be V-shaped. Chromosome will be V-shaped. Metacentric chromosome will be V-shaped because spindle fibers are attached to centromere and they start pull them towards the respective poles. And then submetacentric is the centromere is displaced to the other side of the chromosome, where double the length is here, and then this is one length, and this is a double length. And this is L-shaped structure that you get. L-shaped. This is V-shaped. This is L-shaped structure you get at the time of movement of chromosomes during cell division. And then acrocentric is you have almost to the end a very small piece of chromosome is there, and this is a centromere. And this would be almost a smaller type of L shape. And the last is telocentric, where the shape is I-shape because centromere is towards the end of the chromosome. So you have 3 kinds of situations here where the centromere will divide, will be responsible for the shape of the chromosome at the type of chromosomal movement. Okay, so this is metacentric, V-shape, submetacentric and acrocentric should be L-shape, telocentric should be I-shape. So in the exam, they might ask you what is the nature of alphabet that you have in case of telomere at the end, then you should be able to answer that. Yeah. And then there are some special kinds of chromosomes also. The one that we have seen is a typical chromosome, but in addition to them, we have some special kind of chromosomes also. 1881, E.G. Balbiani first observed polytene chromosomes. This is very important. Polytene chromosomes, this is very, very important. They were found in Chironomus. This thing, Chironomus is the thing that we have polytene chromosome. Which part of polytene chromosome, which part of the cell? They are present in salivary glands. This is very important. Polytene chromosomes are present in salivary glands of the larvae that are there. So they have a lot of salivary glands. Now the question is, why should they have polytene chromosomes in salivary glands? See, larval stage is the most voracious eating stage. It goes on eating the leaves, nothing else. So it needs more saliva for digestion. It needs more saliva for the material that is being ingested to be digested. So that is the reason why larval salivary glands are included under polytene chromosomes. So this was first reported in chironomas. Then we have lamb brush chromosomes also. This was observed by Fleming, and the name lamb brush was given by Ruckert because it looks like a brush which is used to clean a lamb. Usually these are found in the fishes, amphibians, reptiles, and birds, mostly in the oocyte nuclei. Okay, they are present only in the oocyte nuclei, and usually you can observe them in diplotin phase. So that is what we find them. And then B chromosomes or supernumerary chromosomes are also there. These were first reported by E.B. Wilson. These were found in Tradescantia, in insects, in maize, in animals, and then B chromosomes are also found in all tissues of that. So these are special kinds of chromosomes. Okay. So this is the polytene, this is the lamb brush, this is the B chromosome. The polytene chromosomes are seen in case of Drosophila. Okay. So this is the oocyte, and these are the chromosomes that are present with the B chromosome. Yeah. One more important issue is that Rio discolor has ring chromosomes. In the exam, they will ask you where are ring chromosomes present. So Rio discolor has ring chromosomes in its polar mother cells. We can isolate them in the laboratory and then also study them under a microscope. Okay, right. So this is the information about chromosomes that we know. The second part of the information that we know is chromosome number of a plant or an animal or a species is constant. It will not change. Chromosome number is constant for a particular plant or an animal or any living organism. Okay. Now, why you say this chromosome number is constant is what we see here. For example, take a plant called Chlamydomonas. Chlamydomonas is a very small algae. Okay. It is a haploid. It is not diploid, and chromosome number is 17. Then Spirogyra is 4. Yeast is 16. Mucor is 2. Mucor has 2. So these are haploids. And then in plants, Ophioglossum reticulum, 2n is equal to, this is diploid number. 2n means diploid number, 1272 chromosomes. Then Haplopappus gracilis is 2n is equal to 4 number of chromosomes. Then Arabidopsis thaliana, 2n is equal to 10. And then you have animals. You see, for example, elephant has 56 chromosomes, but there's no relationship between the number of chromosomes and the size of the animal. There is no relationship between the number and the size of the animals. Drosophila melanogaster has 8. Then the Culex pipens has 6 diploid number. And there is also a suggestion that dinosaur, which is a fossil, would have 80 chromosomes. A diploid would have 80 chromosomes. So the chromosome number of an individual species is constant. It will not change. Okay. So this is one very important issue. So these are the pictures. This is Mucor, Haplopappus, Ophioglossum. These are the Drosophila. This is the your African elephant. You have the dinosaur. This is a Pisum sativum. This is the Mucor that we think of, and this is Arabidopsis. So their number is constant. The number don't change. And there is no relation between the number of chromosomes and the size of the organism. The organism size can be small, but the chromosome number can be more also. Okay, right. There are 2 types of chromosomes. First is called autosomes. The other is called allosomes. What is the use of autosomes? What is the purpose of having autosomes? Autosomes are chromosomes which carry body traits. That is body characters. Body traits or body characters. While allosomes are responsible for gender, that is male or female discrimination. Okay, whether the individual will be a male or individual will be female, that is done by only two chromosomes. So genes that are present on the 2 chromosomes, right? And the numbering in case of autosomes is usually 1, 2, 3 to 22 in case of human. But if it comes to allosomes, the alphabets X, Y, Z, X, Y are used, and in other organisms Z and O are also used. So Here it is Arabic numerals for autosomes. Here it is alphabets, English alphabets for allosomes. Then genome number is very high. The number of chromosomes, the number of autosomes is very high, while the number of allosomes are usually 1 or 2. And autosomes always occur in homologous pairs. Please make a note of this. Autosomes always occur in homologous pairs, while allosomes are non-homologous. For example, take X and Y. I can show you a picture in the next picture. This next picture shows that Y is smaller and X is larger. But that does not happen with autosomes. Autosomes are equal size and they're homologous pairs. So 11 chromosomes pairs are present in case of human. Right. So this is what I was trying to tell you. This is Y chromosome. This is the X chromosome. See, there is variation in size. So they are not homologous. There is no homology between these two chromosomes. Right. What is the use of having autosome allosome studies? This is responsible for studying sex determination. In Homo sapiens, we have XY male, XS female. Okay. And in Leges triticus, it is XX, XY, XX. In fowls and reptiles, it is ZZ and ZW. In nematode, that is Caenorhabditis elegans. Yeah, one very important thing here is, it is the autosome versus X chromosome. The sex differentiation in Caenorhabditis elegans is X is to A if it is 0.5 and then 1. This ratio decides which is male, which is female, or which is hermaphrodite. Then insect progenitor also has XX and XO. So these chromosomes help in sex determination. Yeah. Then another important aspect of chromosomes is ploidy status, as I've already shown you. Haploids, diploids, polyploids. Polyploids can be triploids, tetraploids, and hexaploids. All of them deal with chromosomes only. Triploids are found in watermelon. Potato is a tetraploid. Wheat is hexaploid. Algae, fungi are usually haploids because they have only one set of chromosomes. And most of the cultivated plants are diploids. Okay, so I can show you them. See, this is wheat. It's a it's a hexaploid. Wheat is a hexaploid. So you have the genome AABB that is present in this polyploid. And then you have triploids. Watermelon is a triploid. So 3 sets of chromosomes are there. And yeah, another important aspect of the chromosome is in tissue culture through anther culture, we can produce haploids. We can produce haploids, and haploids will be diploidized to get a dihaploid. So haploids are found in the plant system in pollen mother cells. So those can be utilized for producing androgenic haploids through tissue culture. Yeah, I've something to tell you about Pomato. Pomato was a theoretical purview of Melcher's. What he decided was that he wanted to fuse 2 protoplasts, protoplast of potato and protoplast of tomato. And his idea was that the aerial parts will produce tomato, the underground root system will produce tubers. That was what he was trying to study. And then he was not successful in this. One main reason for not being successful in this is there is no homology between the chromosomes of potato and tomato. For example, I have given you a picture here in which we say that potato chromosome number is 48, while tomato chromosome number is 24. So there will be no homology between these chromosomes. If there is no homology between these chromosomes, it will be sterile. Usually it will be sterile, right? So theoretically, heterocheron did not survive because of lack of homology in potato and tomato chromosome. This could be one reason, maybe there are other reasons. So we are not able to get a hybrid of potato and tomato, which we call as potato. So again, the reason for this is chromosomes, right? Yeah, this is what I am trying to show you. Here you have a tomato, potato, eggplant, that is brinjal, pepper. All these have a different kind of chromosomal arrangements where we have translocations and inversions have taken place during evolution. So definitely there will be no homology between potato and tomatoes. It will have to be resolved. This has to be first resolved. So that is one reason why pomato has failed, because of there is no homology, or we call it as loss of synteny. This is also called loss of synteny. Okay, you want to see chromosomes. What do we use? Is a microscope. High power is 40x, oil immersion is 100x. Okay, here students are trying to watch them under a normal microscope. This is usually what happens in ileum. This is ileum roots. You have chromosomes here. Okay, you have chromosomes being distributed at telophase, and then you have here late metaphase. This is a prophase. So this is what we see in case of ileum roots. This is animal cell culture. In animal cell culture, you have these kinds of appearances under a microscope. Yeah, this is same again. Root tip. Root tip is the best material for studying mitotic chromosome. So at 4x you see the root tip like this. At 10x you see the chromosomes like this, and 40x you see the chromosomes. These are chromosomes which are early, late metaphase and early telophase situation. See, this is what happens in case of pollen mother cells. This is what happens in case of root tip cell. This is called squash method. You call this as squash method. Root tip mitotic study is called squash method. Then pollen mother cells is called a smear method. In squash method, we use arsine, that is acetoarsine. In smear method, we use acetocarmine. Both of them are observed under a microscope. But in first case, we use arsine. Second case, we use carmine. And the first case you call a squash method. The second case is called a smear method. So in exam, they might ask you, what is a squash method, or squash method is usually studied there, that they can ask you, right? In addition to them, in addition to your regular, we also have other staining methods. Staining studies are there, like DAPI staining. DAPI is 4,6-diamidino 2-phenylenediol. Okay, so DAPI method is also used for staining chromosome. Fish method is another method where fluorescent probes are used, fluorescent dyes are used, fluorescent in situ hybridization. This also we need to have a fluorescent microscope for which we can observe. So these are the new techniques that are used to stain chromosomes. Okay, so this is how it appears. DAPI stain appears like this. This is fish staining. See, the chromosomes are stained. This is centromere, and these are bands. So you have telomere. So different types of stainings are also developed. Okay, right. Okay, using these chromosomes, we can also develop karyotypes. Okay, so this is a male karyotype. This is a female karyotype. So X and Y, XX. So you can develop a karyotype. In karyotype, usually it starts from large size to small size. In humans, 1 chromosome and then 22 chromosomes. So large size to small size. Okay. What is the use? I'll tell you in the next. Okay. This is human ideogram. This is how we develop a human ideogram. This is a centromere. Centromere is placed in the center, and you get a banding pattern in case of ideogram. Okay. Right. This is called aminosynthesis. This aminosynthesis was actually originally used for prenatal diagnosis to find out whether the chromosomes have some mutations in them, some inversions, some deletions, etc. So this was very much useful earlier for karyotype analysis. As I've already shown you, this karyotype analysis can be done by aminosynthesis. Okay, and then as a result of aminosynthesis and karyotype analysis, scientists have found out these diseases. Number 1, Turner syndrome. Either it is 45X. Okay, Klinefelter syndrome, 47. 1 chromosome increased, 1 chromosome is lost. Edwards syndrome. Okay, Down syndrome, Patau syndrome. What do they tell us is that either a chromosome is increased or mostly chromosome is lost. So aminosynthesis and karyotype analysis will help us to find out whether there is disease or not, or syndromes are visible. See, this is one example. We can see Turner syndrome here, loss of chromosome. Then Klinefelter syndrome, addition of 1 chromosome. So Turner syndrome and is 45. Then Klinefelter syndrome is 47. Yeah, and then usually what happens is the ends of the chromosome is called telomere. In this telomere region, usually during replication, a small piece of telomere is telomeric region is not replicated. As a result, what happens is it results in senescence. it results in aging, it results in cancer. Okay, chromosomal study will help us to find out whether the telomeric region is replicated or not, right? One very important observation is that during 2020 corona pandemic, most of the men succumbed to COVID-19 virus. You see a lot of coffins are there. Most of them are men. Women also died, but most of them are men. Scientists have found that X chromosome genes may augment immune system and thus nullify or reduce the effect of viral diseases, COVID. Hence, men fall victim to COVID-19 and other factors like smoking, drinking also elevate this problem. X chromosome is present in women twice, but in men it is only one. So maybe this augmentation of immune system by X chromosome is not sufficient. Hence, a lot of men died than compared to women. This is pertaining to chromosome. And this story that we know is about the Human Genome Project, HGP. I will not go into those details. All the 22 chromosomes, X and Y chromosomes have been sequenced. We know which gene is present on which chromosome that can be taken up, downloaded from Human Genome Project. Okay, this is a Human Genome Project. Now it helps us in disease diagnosis. It also helps us in detecting genetic predispositions to diseases. All these things can be worked out from Human Genome Project. And then chromosome walking is another technique that is used to sequence the DNA. Mostly the genomes of mouse, zebrafish are also being worked out using chromosome walking. This is how chromosome walking is done. I will not go into those details. Students will become for them, right? Okay. One important question is, why do we use garlic or onion roots for squashes? Basically, 2n is equal to 16. So chromosome number is, n number is 8. Chromosomes are very large. This is one very important thing. Chromosomes are very large in onion roots. The 10⁻⁷ times more than Arabidopsis. 83% of DNA is repetitive DNA. Therefore, it is very easy for us to work on root squashes of onion or garlic. So in exam, they will ask you, why do we use garlic? Then you should be able to tell that because of large size of chromosomes. So this is how the chromosome prophase, metaphase, anaphase, and telophase is what appears. Okay. And then second important thing is chromatin. This is the difference between chromosomes and chromatin. Chromatin means the inner material composed of nucleosome, which is exactly like this. So this is DNA, these are proteins, these are loops, okay, these are condensed, and these are chromosomes. So this material is called chromatin material. So we know that DNA is 2 nanometers or 20 angstroms. Then the second compaction is 11 nanometers. Third compaction is the second compaction is 30 nanometers. Third compaction is 300 nanometers. Fourth compaction is 700 nanometers. And finally, the size is 1400 nanometers in case of chromosome. So this is what we call as chromatin material. The material, the DNA material, the protein material that is present inside the chromosome is what we call as chromatin material. Okay. So this is the type of structure, double-stranded DNA, DNA plus nucleoprotein. The next level of compaction is fiber. Then you have loop, and then we have chromatids. Okay. So this, it is said that if all the chromosomes in humans are attached head to head, it will be a length of 2 meters. And this 2 meters DNA present in the 46 chromosomes are present in 5 to 10 new micron nucleus. So compaction should be very, very important if it has to be compacted or packed into a nucleus of this type. Okay. So this is euchromatin. This means genes, heterochromatin, non-genes. They don't, these are silent genes. These genes are not functioning, but these genes are functioning. Euchromatin genes are functioning, while heterochromatin genes are not functioning. 2 to 3% of the genome is euchromatin. 97 to 98% of the genome is heterochromatin. Okay, but there is one advantage. I'll tell you what that is. This advantage is this heterochromatin has helped Alec Jeffrey, who developed DNA fingerprinting. What is DNA fingerprinting? DNA fingerprinting is based on heterochromatin material. Some of these heterochromatin material are called mini satellites. They are called mini satellites. So these are silent. These mini satellites have come handy for Alec Jeffrey, and using this technique, he has developed one technique called DNA fingerprinting, and DNA fingerprinting has good applications, forensic science. You can paternity testing. You can also use in medical diagnosis, agriculture, food industry, sociology. Okay, that is identification of racial groups. Okay, migratory people. That also can be found out in case of DNA fingerprinting. So DNA fingerprinting again has basis on chromatin material, and in chromatin material, as I've told you, chromatin material is very important in terms of for this technique. And this chromatin material, all the DNA were active at once, but now they become silent, or they become silent genes. Okay, right. So I request all the students who are interested to appear for these exams to follow these books, Botany and Biotechnology. Editors were Professor S.M. Reddy sir and Professor Ram Reddy sir. And 5 of us have authored this. You can get these books from Telugu Academy. Also, you can have P.K. Gupta Genetics, Verma and Agarwal Genetics, P.K. Gupta Elements of Biotechnology, H.S. Chawla Introduction to Plant Biotechnology. These you can read. Telugu Academy books are very good. You can use them. There is no doubt about this. All the students can go through them. They are very simple, but very informative. So you can use those books for studying for the entrance test. Okay. So thank you very much for your patient hearing. Any questions? I'll be able to take. Hello, Tirupati?
Yes, sir. Yes, sir.
Any questions? Anybody?
Yes, all the participants, please, you have any queries or any doubt regarding of CPGET or PG entrance or any other books, please you interact with our professor sir. Our esteemed professor sir is here. All of you, please. మాట్లాడండి, మీరు మాట్లాడొచ్చండి.
Christopher sir, thank you so much for your valuable lecture on this platform. Participants, please, you can interact with sir for any queries.
Anybody else?
I hope nobody, sir. All the topic what sir has said, those all are important for the exam point of view, sir. All the points will be covered.
Excellent.
It was an excellent.
Tirupati, okay?
Sir, really, thank you very much, sir.
Okay.
Kavita, mana Rajini madam garu vote of thanks.
Yes, sir. Yes, sir.
Invite cheyandi.
Yes, sir. Sir, thank you very much, sir, for your valuable presentation on this platform. This was a very, I hope our participants have got the knowledge on, a small knowledge on cell biology in the exam point of view.
Yes.
Thank you very much, sir, for accepting our invitation.
Okay, madam. Thank you so much. See you again, madam. Tirupati,
Thank you,
bye.
Sir.
Thank you, sir. Now I invite Dr. A. Rajni, Head Department of Botany, RBR Women's College, to speak few words and propose vote of thanks.
Good afternoon, one and all. I welcome you all for National Online Coaching, Plant Science and Plant Biotechnology. It's my pleasure to welcome today's chief guest, Dr. Sudarshan Reddy sir, General Secretary, Telangana State, keynote speaker, Professor T.Christopher sir, Department of Biotechnology, Dean, R&D, CDU, Varangal. Inaugural talk speaker, Professor V.KRISHNAREDDY sir, retired professor, Kakatiya University, Varangal. I also invite all my Professor Sudarshan Reddy sir, Secretary cum Correspondent, RBVRR Women's College, Dr. Achyuta Devi, Principal of the RBVRR Women's College, Dr. C. Kavita, Vice Principal of the RBVRR Women's College, Dr. Y. Satyanarayana Sir, Principal of the GDCW Jagitial College, Dr. T. Sri Lakshmi, Principal of the GDCW Karimnagar, Dr. K. Ramakrishna, Principal of the SRR GASSC Karimnagar. I also all my dear colleagues and student friends. Now I would like to brief you all about our department. Department of is established in 1954, and we have introduced food and nutrition as one of the optional subject at UG level in 2014. Department is supported by 6 faculty members, which are PhDs holders, and 1 are pursuing PhD. Department has 4 lab and well-maintained museum, which rare collection of herbarium. Department has the credit of having a tissue culture lab. We have 6 MOUs, 4 linkages, and we also have Eco Club, in which we conduct various student activities. Department regularly conducted seminars, workshops, extension lectures, and field trips to update student knowledge. We also take up student project at UG level. It gives me delight to express the glorious and successful completion 2 years online coach and step in towards the 3rd year, hopefully desired to achieve 100% result from the students by screening top most rank in all the universities. I hope students gained immense knowledge to do today's national online coaching. The keynote speaker enlightened to students on life sciences, which is beneficial in appearing for the PG entrance examination. All the best, students. Thank you, sir.
Okay, thank you, Rajini Madam Garu. So we are wholeheartedly thanks to each and everyone, and along with our co-patrons and patrons of today's program, and the most eminent personality, Dr. Kadaru Surendereddy Garu, General Secretary TGCGTA, today's chief guest. So because his encouragement, we are still continuation of our programs forever, along with our collaboration with RBVR Women's College, SRR Government Arts and Science College Karimnagar, and Women's College Karimnagar. So we are still continuing for our what we are desired, we're going to fulfill with the students' participation and encourage the students' participation. We are wholeheartedly thanks to each and every resource person, those who are going to involve for tomorrow onwards, and also in previously who worked, who sincerely served as a resource person in previously. We are wholeheartedly thanks to each and every resource person and previous 2022 CPGET 2022 and also CPGET 2021 also resource person. They are given their valuable time and they are spare their knowledge and share their knowledge among the students, and the students, they are already get benefited, joined different universities in all over Telangana and also other states, especially in Andhra Pradesh. Shivaji Nayak was the among 11 universities of Andhra Pradesh. He got first rank in 2022 in Andhra Pradesh. So just because of this coaching. So in previous year, we are on the banner of TGCGTA. We are successfully conducted. And at the same time, this year also, we are also same platform and the same banner we are using for the will going to successfully complete our program. Thank you, Rajini Madam Garu and Kavita Madam Garu, and being a part of even the entire 65 days uninterrupted program, and also the resource persons will going to collaborate and coordinate with combinedly or conjointly continuous our programs, our classes will going to finalize. Okay, thank you very much, madam. And also Surendereddy sir, really thank you, sir, for your valuable time. The most probably 2.5 hours you are giving your your valuable time, sir. And also my professor also already sir reminded you, sir, because of your great efforts and strong determination is really inspirable, sir. Thank you very much. With your permission, we are going to wind up, sir. If any words, any few words, please you interact, sir. Otherwise, please, we will going to wind up, sir. Dr. Surendereddy, sir.
Sir, I think sir has left. Sir is not there in the meeting.
I am sorry
Sir,
to say this.
you're mute.
I am sorry to say this is a time because of Dr. P. Manjula Madam Garu has given her valuable services last year to CPGET 2022. Really, my bottom of my heart, really sorry. Please you accept my what I am saying here on this platform. Really, but I am asking about your photograph. Genuinely, I am saying our head department of
Ma'am is Manjula is not there now at present in the department.
Manjula is not there, but this video is going to reach our Manjula Madam Garu. So that's
okay. Recording.
I'm saying my own words. Really, Madam has given valuable time and also express her knowledge. And previously, more than 25 days, Madam has continuously given her valuable time. So that's why I wholeheartedly thanks to our resource person and a great personality, Dr. P. Manjula Madam Garu, Assistant Professor, Department of Botany, RBVRR Women's College, Autonomous, Hyderabad. Thank you very much. Okay, I hope you please you convey.
Okay, sir. Okay, sir. Definitely will convey.
Okay, thank you. And also my beloved friend, my classmate, Dr. A. Rajini Madam Garu and Kavita Madam Garu. Both of they are really supporting me for continuation of this program. This is a wonderful program because of in entire Telangana state, nobody has ready to continue this much of pain and also ready to give the services towards the students' benefit. So really hats up to you and also the principals of our all colleges. Wholeheartedly thank you. With your permission.
Thank you, sir.
Sir, this credit not to our sir. This credits goes to our sir Vishwanathan sir.
Sir?
Credits not to me and me, yours, ours, sir. These credits go to our Vishwanathan sir.
And also the one person, secretary and correspondent of RBVR Women's College, Professor Sudarshan, also really his encouragement is nothing but a boost of our energy.
Definitely.
For giving giving of our additional time for the sharing of our knowledge. So really thanks to each and everyone. We will going to wind up. So tomorrow onwards, my dear friends, tomorrow morning, maybe I will sharing the schedule, sharing of our WhatsApp group. Please you follow the timetable. Okay. Thank you.
Thank you, sir.
Thank you.